name is Gunvant Patil, and I'm associate professor at Plant and Soil Science Department, and it is also a part of like Institute for Genomics for Crop Abiotic Stress Tolerance. So my lab works on like two major areas. One is like trait discovery, where we try to integrate like a genomics and genome engineering technologies to discover new traits which are important for crop improvement, for example abiotic stress or biotic stress. On the other side, like one of the major area that we are also working on developing the new technologies for improving transformation or improving the regeneration, transformation and gene editing in some of the recalcitrant crops including sorghum, soybean and cotton. On a region where we grow a lot of cotton, sorghum and different crops, and I know like what are the importance of the semi-arid regions like West Texas, where there is a water scarcity. So that always inspire me like how we can improve the crops to have like a better water use efficiency, nutrient use efficiency, and how these crops can combat against the pathogens which are present in the environment. So that's like the pressing needs that farmers always struggle with. So to improve those, I started my education in like basic botany, and then I studied like more about the plants, and that again my more interest to study plants at not only the genetic level but also at the molecular level, and that keeps me inspiring. Like there are like lot of new traits, not lot of new genes that we can discover, and these genes can be used for improving the crops for the regions like West Texas, where there is a poor soil, water is scarcity, and there is like a lot of heat, and also at the same time there is a lot of pressure from insects and different diseases. So that those challenges always inspire me to work on those crops, use my understanding in genetics, genomics and genome engineering to improve them. As I mentioned, like my lab focus on improving the technologies for genome engineering. So one of the focus of my lab is like we can improve the transformation or like develop a technology where we can accelerate the regeneration and transformation in sorghum. So to improve any crops through the genetic modification or genome engineering, the regeneration and transformation is the one of the major bottleneck. And that is something what my lab is trying to do to improve that technologies. So once we have a reliable technology to transform recalcitrant crops, this technology can be utilized by students for discovering new traits, new genes. At the same time, once we have the reliable technology or the protocol for improving transformation, this technology can be used indirectly by the breeders and the industry to improve particular trait. So basically what my lab does is like we try to improve these technologies and modify this technology to streamline the process so it can be easily adopted by students for a basic research, as well as it can be adopted by the industry for integrating the new traits into the crops. Yes, so my research is heavily involved, or like I heavily collaborate with different groups including students. So students are the backbone of my research. They are the one who get like training to develop these tools and technology in the lab. Also you implement this technology for the trait discovery. So definitely students are the backbone of my research. Additionally, I collaborate with different industry including the commodity boards who are like also provide us funding support to perform the research, and those resources we utilize for improving our like gene discovery platforms, use those to improve the technologies that we have. And once this is like a mutual benefit from the commodity like that provides us the funding or resources, and in return we provide some technologies that can be utilized indirectly to these communities. Additionally, I also collaborate a lot with the federal agencies like USDA or a National Science Foundation, and those fundings are very important for my research. And finally, there are like some industry partners like the seed companies. Those are also a big, they play a important role in my research by providing the resources and fundings. Yeah. So as I mentioned, like my lab focus on genome engineering, and one of the major challenge in the genome engineering is like the transformation bottleneck. For example, if you are working on sorghum is very difficult for regeneration and transformation. So those are like some challenges like genotype dependency. It requires a long time for regeneration or transformation. For example, sorghum, if you start an experiment, it takes at least 1 year to get your transgenic or gene-edited crops, gene-edited sorghum plant. And therefore, my lab is trying to minimize those duration and trying to make a very efficient process, so we can not only reduce the time, we can also improve the efficiencies across the different genotypes which are important or which are established for the West Texas and which are specific to that particular area. So altogether, like my research focus on reducing the bottleneck associated with recalcitrant nature, genotype dependency, reducing the time for developing the engineered crops, and also making the process more efficient. So the next, so since we are working on developing these technologies to improve regeneration and transformation or overall genome engineering in sorghum, so far we have made a lot of improvement in these methods. For example, we were able to reduce the regeneration and transformation time in sorghum by almost like halfway. Like for example, traditionally it requires about 7 to 8 months. Now using the new methods or new technology that we have developed in my lab, it can regenerate a sorghum plant within like 4 months or less than 4 months. So that's like one of the key achievement that we have. One thing I really would like to highlight is like so far conventionally researchers have shown that sorghum can be transformed, only 1 or 2 genotypes can be transformed efficiently in sorghum, but through our research we can show that we can transform multiple sorghum genotypes, not only for grain sorghum, but we can also do the transformation in the forest sorghum or bioenergy sorghum very efficiently. Yeah, one thing I would like to mention that crops can be improved by different technologies. Of course, like genetics is one of the important technology or tool to improve the crops. Additionally, plant physiology, agronomy, and so on. But in addition to that one, the genome engineering or genome editing is one of the new technology or emerging technology that can be also utilized for crop improvement. The genome editing is like another important technology that can be utilized not only to discover the new traits or new genes, but this can be a technology where we can precisely make any modification in the plant genome. So in nutshell, I would like to mention that considering like more technologies and tools available in the plant biology and overall agriculture, genome engineering technology which I'm working on and which we are trying to improve is going to play a very important role in agriculture for any given crop.